बिस्मिल्लाहिर्रहमानिर्रहीम। इस वीडियो में हम जेनेटिक कोड के इंट्रोडक्शन के बारे में पढ़ेंगे, इसकी डिफिनीशन के बारे में पढ़ेंगे, इनिशिएशन कोडॉन किसको कहते हैं, टर्मिनेशन कोडॉन किसको कहते हैं, सेंस कोडॉन, एंटी सेंस कोडॉन, एंटी कोडॉन, कैरेक्टरिस्टिक्स ऑफ कोडॉन एंड क्लिनिकल सिग्निफिकेंस ऑफ जेनेटिक कोड के बारे में पढ़ेंगे। तो चलिए शुरू करते हैं। फ्लो ऑफ जेनेटिक इंफॉर्मेशन। जैसा कि आपको मालूम है कि डीएनए बनने के अमल को डीएनए रिप्लिकेशन कहते हैं, डीएनए से आरएनए बनने के अमल को ट्रांसक्रिप्शन कहते हैं और आरएनए से फिर प्रोटीन बनने के अमल को ट्रांसलेशन कहते हैं। इस प्रोसेस को हम सेंट्रल डोग्मा ऑफ मॉलिक्यूलर बायोलॉजी और सेंट्रल डोग्मा ऑफ मॉलिक्यूलर जेनेटिक्स भी कहते हैं। द सिंथेसिस ऑफ डीएनए फ्रॉम डीएनए इज नोन एज डीएनए रिप्लिकेशन, द सिंथेसिस ऑफ आरएनए फ्रॉम डीएनए इज नोन एज ट्रांसक्रिप्शन and the synthesis of protein from RNA is known as translation. So the information is always from DNA to RNA to protein. The pathway of protein synthesis is called translation. जैसा कि आपको मालूम है कि प्रोटीन बनने के अमल को आरएनए से हम इसको ट्रांसलेशन कहते हैं, because the language of nucleotide sequence on messenger RNA is translated to the language of amino acid sequence. You know that in messenger RNA, a specific sequence of nucleotide is present, from which specific sequence of amino acid in a protein is produced. So the process of translation required a genetic code, through which the information contained in the nucleic acid sequence is expressed to produce a specific sequence of amino acids. Then what is genetic code? फिर genetic code कैसे कहते हैं या हम इसकी definition किस तरह करेंगे? Genetic code we can say three adjacent, for example UCNG or CUNA, three adjacent nucleotide on DNA or on messenger RNA is constitute are called genetic code. Genetic code is a set of rules by which information encoded in genetic material in the form of DNA or RNA sequences is translated into protein by living cell is known as genetic code. The word genetic code for the first time given by George Gamow. The discovery of genetic code. You know that to understand how proteins are encoded begins after the structure of DNA was discovered by James Watson and Francis Crick. जिस तरह हमको मालूम है कि DNA की स्ट्रक्चर सबसे पहले जेम्स वाटसन और फ्रांसिस क्रिक ने 1953 में दिया था. George Gamow postulated that a three-letter code must be employed to encode the 20 standard amino acids used by living cell to build a protein. So the word genetic code for the first time used by George Gamow. The later adenine, guanine, thymine and cytosine correspond to the nucleotide found in DNA. They are organized into codon. What is codon? The collection of codon is called genetic code. For 20 amino acids, there should be 20 codons. Each codon should have 3 nucleotides. We simply define genetic code 3 adjacent nucleotide on RNA or DNA is known as a genetic code. So 3 nucleotides to impart specificity to each of the amino acid for a specific codon. So when one nucleotide it have 4 combination. When we use 2 nucleotide it have 16 combination. And when we use 3 nucleotide, so it produce 64 combination. So this 3 nucleotide genetic code 64 combination most suited for 20 amino acids. So you know that 20 amino acid is responsible to produce various types of protein from bacteria to the human beings. So genetic code will be composed of 64 codes. This is the genetic code. You know that the synthesis of RNA from DNA is known as transcription and from RNA to protein into the language of amino acid is known as a translation. So 3 adjacent nucleotide is basically called a genetic code. Genetic code can be expressed in a simple table with 64 entries. This will be first position or first letter, this will be the second position or second letter, and this will be the third position or third letter. For example, when this U is crossed with the second letter and then to the third letter, it produces triple U, and this triple U genetic code is specific for phenylalanine amino acids. Then WC is specific for phenylalanine, then UAA, UUG, etc. This is for specific for leucine amino acids. In this way, 64 genetic code is produced, which is specific for 20 different types of amino acids. These three codon, UAA, UAG, and UGA, is known as stop codon. From bacteria to the human beings, they are simple. Codon and its type. What is code and what are their types? Genetic code is a dictionary. Consists of genetic words called codons. Each codon consists of three bases. Simply we define code three adjacent nucleotide. Three adjacent nucleotide present on RNA or DNA is known as a code. And you know that there are 64 codes, 61 code for 20 amino acids found in protein, and 3 codon do not code for any amino acid, which is known as the stop codon. Types of codons. Sense codon. What is sense codon? The code that code for amino acids are called sense codon. We know that there are 61 codes for 20 different types of amino acids. Signal codon. Those codon that code for signal during protein synthesis are called signal codon. They either start the process of translation, protein formation, or either they stop the process of protein formation. So AUG, UAA, UAG, and UGA, these are known as signal codon. Three of them they are known as stop codon, and one of them they are known as a initiation codon or starting codon. So there are two types of signal codon, terminating codons and initiating codon. Initiating codon which start the process of protein formation, and terminating codon which stop the process of protein formation. UAA, UAG, and UGA are termination codon or nonsense codon, and often referred to as ember, Ocher, and codon or stop codon. Initiation codon. AUG is the initiation codon. It codes for the first amino acid methionine in all protein. At the starting point, it codes for the methionine in eukaryote and formyl methionine in prokaryotes and bacteria. Anticodon. The base sequence of transfer RNA which pair with codon of messenger RNA during translation is known as anticodon. For example, this is the piece of messenger RNA having these type of nitrogenous bases, uracil, cytosine, uracil, or cytosines. So the anticodon present in transfer RNA is adenine, guanine, and guanine. So you can say that the base sequence of transfer RNA which pair with codon of messenger RNA during translation is known as a anticodon. Difference between codon and anticodon. Codon could be present in both DNA and RNA, but anticodon is always present in RNA and never in DNA. Codon are written in 5 to 3 direction, where anticodon are usually 3 to 5 direction. Anticodon of some transfer RNA molecule have to pair with more than one codon. Codon are sequentially arranged in nucleic acid stained, while anticodon are directly present in cell with amino acid attached or not, or free. Codon defines which anticodon should come next with an amino acid to create the protein strand. Anticodon help in bringing a particular amino acid at its proper position during a translation. What are the characteristics of genetic code? As we have studied some characteristics of the genetic code, some codes are known as start codon, for example AUG, and some codons are known as stop codon or terminating codon. The genetic code is always triplet. The genetic code is triplet mean that each genetic code composed of 3 adjacent nucleotide nitrogenous bases. And you know that there are 64 codes, and within the 64 codes, 61 are known as sense codon and 3 are known as a stop codon. The code is universal from bacteria to the human beings. The genetic code is universal. Each code is specific for their specific type of amino acids. So this is known as universality. Non-ambiguous. The genetic code is non-ambiguous. Thus, one codon cannot specify more than one amino acid. So, this characteristics of genetic code is known as non-ambiguous, non-overlapping. We can say that genetic code is commaless or non-overlapping. One base cannot participate in the formation of more than one codon. This means that the code is non-overlapping. Continuous translation, here we can say that the code is commaless. The gene is transcribed and translated continuously from a fixed starting point to a fixed stop point. So punctuation, for example, comma, etc. is not present between the genetic code. So we can say that genetic code is continuous translation, or we can say that genetic code is a commaless. The code has polarity. The code has a definite direction for reading of message, which is referred to as polarity. Reading a message from left to right and right to left will specify for different amino acid. For example, UUG stand for leucine, and from right to left it GUU which stand for valine. So we can say that the code has polarity. Degeneracy of genetic code. An amino acid can be encoded for by more than one codon, and this is called degeneracy of genetic code. For example, CUU, CUC, CUA, and CUG. These are four different types of code, and these code are specific for leucine amino acid, and UUA, UUG, it is also specific for leucine amino acid. So six different types of genetic code is responsible and specifics for leucine amino acid. So this property or characteristics of genetic code is known as degeneration of genetic code. And this is the flow of genetic information from DNA to protein. And these two process transcription and translation. Clinical significance mutation can be well explained using the genetic code. Mutation means any change in DNA is known as mutation. And there are point mutation, and their example is silent, missense, and nonsense. And the second one is frame shift mutation, silent mutation. Single nucleotide change A to G. Same amino acid is incorporated, mutation goes unnoticed. For example, in this case, GAA, when instead of adenine, when they produce glutamine amino acid. When instead of adenine, guanine is present, mutation is takes place. Then this guanine, adenine, guanine is also specific for glutamine type of amino acid. So this type of mutation is known as unnoticed, and this type of mutation is known as silent mutation. Missense mutation, single nucleotide change A to C, different amino acid incorporated, so loss of functional capacity of protein. For example, here instead of adenine, cytosine is present. So from here his histone protein is produced, and here proline, incorrect amino acid, which may produce a malfunctioning protein. So the protein does not perform their function. So this type of mutation is known as missense mutation. Or nonsense mutation, single nucleotide change from cytosine to thymine. Stop codon is generated and messenger RNA represented by UAG. So premature termination of chain may be incompatible with life. For example, when here a mutation is takes place and the sense codon is converted into stop codon, and further formation of protein or further formation of amino acid is stopped. So this type of mutation is known as nonsense mutation. And what is frame shift mutation? Insertion or removal of a basis can change the reading frame with the resultant incorporation of different type of amino acid. So this type of mutation is known as frame shift mutation. If you like the video, please share and subscribe. Thank you.